So this is moving from the long-term control of the structure of the circulation and the microcirculation to the question of the short-term responsiveness of the vasculature to changes in demands. And this is work primarily carried out with Julia Asiero and Brian Carlson. So just some fundamentals of the regulation of coronary blood flow, which is a very remarkable achievement the blood manages, that in response to changes in oxygen consumption rate over about a 4 to 5-fold range here between rest and maximal exercise, about a factor of 5 is typical in humans. The blood flow, the perfusion in blood volume per time per gram of tissue in this case, goes up almost linearly, and correspondingly the extraction of oxygen from the blood is rather constant in the range of 70 to 80% over that entire range. So that shows you that the flow can vary quite a bit, and yet conversely, if you like, there is the rather tight auto-regulation of blood flow, which is that as perfusion pressure is varied over a wide range, as long as it stays between the range of about 60 to 140, the coronary blood flow is almost independent of arterial pressure. So the question is, how is this achieved? And certainly if as I strongly believe the cardiac physiome should include a consideration of the regulation of blood flow, then we need some underlying concepts of how this works if it's to be incorporated into that framework. So the primary way that blood flow is regulated is in the microcirculation, in the small arteries and the arterioles, which have smooth muscle cells in their walls and can contract and dilate and thereby change the resistance to blood flow. And this is the same picture you just saw a moment ago from Axel. This is the mouse epicardium in particular. And there's a particular problem that arises in thinking about the microcirculation and the control of blood flow. That is that the vessels that supply a given region are not in the same place as the actual region that they're supplying. There's a spatial separation. So in order to have local control of blood flow, you don't necessarily want an arteriole at this place to be responding to its local environment. It should be responsive to what's happening at some remote location. That's shown here. If this area needed more oxygen for some reason, then one would need dilation of all of the upstream vessels here in order to get an increase in supply to that region. So that's the part of the problem. What tools, if you like, what mechanisms does the circulatory system have at its disposal to try to do this? The myogenic response, which is that an increase in pressure causes, or an increase in wall tension to be more specific, causes a contraction of smooth muscle. The shear-dependent response, which is that increases in wall shear stress cause vascular dilation. And metabolic responses, I'm going to focus on the oxygen-dependent response. In general, if oxygen levels go down, that causes vasodilation. And conducted responses, which Axel briefly alluded to, but this is the concept that there are specific mechanisms for transfer of information, especially upstream along blood vessel walls. So I'm going to skip the modeling details to a large extent. This is a rather simplified model in which although much as we work on realistic, complicated network structures, we boil it down to a really bare minimum here, where we consider 7 segments, each one representative of a certain part of a pathway through the circulatory system. And of those 7 segments, these ones are considered fixed resistances, and we're left with these 2, the large and small arterioles. These 2 compartments are capable of active variation of vascular tone. And the question, well, one of the things that we wanted to do in this was to build in a reasonably realistic representation of the mechanics of vascular smooth muscle. And so let's first consider its length-tension relationships. Usually muscle mechanics is expressed in terms of those types of relationships. So in a passive smooth muscle cell or passive vessel wall, there's an extremely nonlinear relationship between length and tension, which usually is fairly flaccid up to a certain point and then very rapidly stiffens up, and that can be well fitted by an exponential function. On the other hand, the active component has an optimum length at which it acts, and that's to do, as you probably know, with overlap of cross bridges and so on in the molecular structure of the muscle, such that there's a peak in wall tension generated at some finite length. And in smooth muscle, that's fairly close to the length at which it becomes much stiffer in the passive response. So the basic idea is that the total tension in the vessel wall can be expressed as the sum of these two components, where the active component obviously is modulated by a quantity A, which can only vary between 0 and 1, 0 being a passive muscle and A being maximal activation. So now the question is, of course, what is A in any given state? And now I'm just going to redefine A. So A goes between 0 and 1, but so just to make absolutely sure that that doesn't get violated, I'll put it into this kind of sigmoidal function that looks like this as a function of another variable, S tone, so that this S tone, the total tone signal, if you like, can vary from -infinity to +infinity without getting us into any trouble. Okay, so now the question is, what determines S tone? And the basic idea is, I think from a modeling framework, this is very important, that we have this one number where the various effects are converging, as they must biologically at some point in the signaling process of calcium release and so on to produce muscle contraction. So we're building in a convergence of those various signals at this point, where we have a term coming from wall tension that gives us the myogenic response, a term coming from wall shear stress that gives us the shear-dependent response. And I'll come back to this one in a moment, but if we put these two in, then we can simulate experiments involving isolated arteriolar segments where the segment is pressurized at different pressures, and either the passive diameter is given, which looks like that, or the diameter where the myogenic response is activated is included. And also we can look at the effect of shear stress induced by flow. And the points here are data points from Akash Koller's group, and the curves here are the fit that the model, including these terms, gives to those data. But now this conducted metabolic signal, that seems to be like the bit of mystery we have to put into this, but it's not so mysterious. It's been known actually for many decades that signals are propagated electrically along the vessel walls via the endothelial cells, and particularly via the gap junctions that form between those cells. I guess for this audience that's not too surprising because everyone's used to that in the context of cardiac myocytes. But anyway, the vessel walls are doing this as well, and so that gives us one piece of what we need. The other piece of what we need is some way of sensing oxygen levels. Obviously, a signal that depends on oxygen level has to have a sensor. And one of the, I think, a big contribution in this area was made by Mary Ellsworth and Randy Sprague and others over the last 10, 12 years, which is to show a very interesting mechanism of sensing of oxygen by the red cell itself. In other words, the red blood cell is acting in this concept not just as a carrier of oxygen, but also as a sensor of oxygen. And it is known that red blood cells release ATP. They have a rather high concentration of ATP inside the cell, and the release of ATP occurs at a rate that is dependent on the saturation of the hemoglobin molecule. So it actually depends on the conformation of the hemoglobin. So as the red cell goes through here, and if you can see it getting a little bit bluer as it loses its oxygen, that would then cause it to release ATP at a faster rate, which can then, according to this concept, and which has been shown experimentally, stimulate an upstream conductive response to the arterioles and thereby cause contraction or dilation of the vascular smooth muscle. And that's expressed here as an integral. This SCR in the previous slide is an integral of the ATP concentration along a flow pathway through the whole tissue, and then with an exponential decay with a characteristic length L0 that we have evidence is of the order of 1 centimeter. So it is long enough for the signals to propagate from the venules right through the capillaries to the arterioles. And I think I'll go over this very quickly, but the main point of this is that the ATP concentration in the downstream segments of the vessel is predicted to go up in the micromolar range, which some experiments have supported. As we change the oxygen consumption rate, the saturation is getting lower. That means more release of ATP and therefore higher ATP concentrations and a higher conducted response signal that's going upstream and reaching these arteriolar segments. So to give some results with this approach, this is for metabolic regulation. It's based on skeletal muscle data rather than cardiac data here. And what it's showing is that if we put the whole model in here, we get something like this, and that the metabolic and shear-dependent responses are actually working against it. In other words, if you add, the myogenic and shear-dependent responses are working against it. So if one has just the metabolic signal, one gets a pretty healthy increase in flow here. But actually, the myogenic and metabolic mechanisms are not contributing significantly and only in a negative way to the metabolic regulation of blood flow, according to that model, which is certainly not something that's the normally accepted concept of how those mechanisms operate. Now, in the case of autoregulation, the situation is maybe surprising, but on the other hand, fairly easy to see what's going on. If you have just a myogenic response, you do get some rather weak autoregulation. Classically, the myogenic response has been considered to be the foundation of autoregulation, but what this would say is that the myogenic response by itself doesn't do the job. And it's quite clear that the shear-dependent response is going to work against autoregulation, because if you increase the arterial pressure, that's going to increase the pressure gradient, the wall shear stress, which is going to cause vasodilation, which is exactly the opposite of what you want for autoregulation. So myogenic plus shear is up here, but then when we add in the metabolic response, then that brings it into a reasonable range. So to conclude, in the microcirculation in the heart, as in other tissues, there's these two problems that nature or biology has to solve of supplying the needs of the tissue in proportion to the needs, but independent of the driving pressure, because the heart can go wrong and you've still got to give it a chance for survival, so, or you can lose some blood or whatever. So how is autoregulation achieved? Autoregulation requires, there's a long debate in the literature, is it metabolic or is it myogenic? What we would say is both are needed, because they have to defeat the shear-dependent response, which is working in the wrong direction, and that this ATP and conducted response mechanism is a plausible candidate for the mechanistic basis for metabolic regulation of blood flow. And also one point I'd like to make is that as in developing these models, it's been an aim to build them in a way that could be incorporated into the framework of the cardiac physiome project. Thanks very much.
Questions? Yeah.
Hi, thanks for talking. So I was wondering, with this conducted response that you're describing, how fast is that process? And also, you described with the ATP diffusion, is that right? And how does that sort of jive with theories of electrical and calcium wave propagation?
Yeah, so the story with conducted responses is getting more and more complicated. There's actually at least two, maybe three independent mechanisms. There's a depolarization or hyperpolarization mechanism, and I think that there's numbers in the range of 1 millimeter per second for the velocity of propagation of that. There's also a calcium wave dependent mechanism, which is I think propagated a lot slower than that, but has been observed to propagate through multiple endothelial cells. So yeah, there is quite a bit of information on that.
Tim?
Tim, in some, lot of tissues actually, the arteriole comes in and the two veins go out beside it, the vena comitantes. And the separation distances at arterial diameters of around, say, 15 to 80 microns, they're very short between the arteriole and the venule. Could that diffusional exchange from the venule to the arteriole be an alternative route compared with the ascending communication through the endothelial cells?
Yeah, and it almost certainly is, and there's quite a bit of evidence to support that. One point to realize, though, is that occurs at a relatively large size. of arteriole and venule. In other words, the arteriole that's paired with the venule is supplying a pretty large region, and the venule that's next to the arteriole isn't necessarily coming from the region that is being supplied by that arteriole. So that can give a fairly broad scale feedback mechanism for regulating blood flow, but it doesn't give the fine scale regulation of blood flow on the level of the individual microvascular units that would obviously give better matching between perfusion and requirements.
Do you have a quick question?
Yeah, is there also neuronal regulation of vasodilation that occurs through?
Yeah, absolutely. And again, my response is more or less the same, and I should have given justice to all of the different mechanisms. But once again, the neuronal regulation does not have this fine spatial scale that allows it to give regulation on an individual level down to the terminal arterioles. So certainly that, obviously neuronal mechanisms are extremely important in blood flow regulation. Yeah, I don't want to minimize that.
Thank you very much.